the primary lymphoid follicle will normally not be exposed to antigens. There's aggregates of lymphocytes, but it lacks the germinocenter. Germinocenter is where we have activation of the lymphocytes. So that's where we have the immunoblasts being formed, and that's where conversion of B lymphocytes to plasma cells, the acquisition of the receptors, and to be able to recognize antigens, that occurs within the lymph nodule. We'll be looking at it later on. So the lymphoid tissue can be either diffuse, or I talked about it, or follicular or nodular. In the areas where you have diffuse lymphoid tissue, you normally have high endothelial veins. I think we mentioned that. Oliver, you can either mute or get out of the class. So the secondary lymphoid tissue, secondary lymph nodule, I've said, consists of germinocenter as the distinguishing feature, and then the mantle zone. I think we had looked at this one where we are looking at a nodule comprising of closely packed cells, but lacking a germinocenter. This before exposure to antigens. And then in the case of the secondary, we have a very distinct germinocenter, and then the germinocenter is surrounded, which are very closely packed, that we refer to as the mantle zone or the cape. And we mentioned that the cape is usually thickest towards the sinus. That's where we have a thicker part of the cape. And in most cases, when you look at this, the outermost area of a lymph nodule comprises reticular fibers, closely packed reticular fibers that form something like a capsule around the nodule. We have finished the lymph node, so let's look at the spleen. I think that's where we are supposed to start today. The spleen, as you'll be aware, is associated with the greater cavity of the stomach. And the spleen, histologically, if you cut a spleen, comprises of white spots, and these white spots are what we refer to as the white pulp, and then the red pulp. The red pulp because it contains a lot of blood vessels. The functions of the spleen, one is quality control in terms of cleaning up the blood. This occurs within the splenic cords. We're going to look at the splenic cords and what happens during this passage of blood within the splenic cords is mainly destruction of 8 deformed red blood cells within the spleen. These 8 or deformed red blood cells cannot squeeze through the spaces, and therefore they are recognized as foreign and are destroyed by macrophages. So the spleen is sometimes referred to as the graveyard for red blood cells. Blood filtration, this also occurs within the cords, removal of foreign proteins within the blood, and also trapping of blood-borne antigens. All this occurs within the splenic cords of Billroth. We also have what we refer to as red blood cells pitting, and red blood cell pitting involves removal of inclusion bodies, for example, hen's bodies or Howell-Joel bodies. The malarial parasites can be removed from the red blood cell without destruction of the red blood cell. So that's what we refer to as pitting of the red blood cell. And this occurs within the splenic cords of Billroth. That is part of the quality control of blood at quality, ensuring that foreign material within blood are extracted and destroyed. We also have storage of plasma, and this occurs, and red blood cells. This occurs within the splenic sinuses is where we have a lot of blood storage, and that acts as a blood reservoir, so that when the blood is needed, for example, after excessive bleeding, this blood can be squeezed back into circulation. That's what we refer to as a blood reservoir. So we're going to look at how is this possible that we have this reservoir being put back into circulation. We also have storage of platelets in ready form within the splenic cords. So platelets are stored, and when there is excessive bleeding, these platelets can be recruited back into circulation so that it can be able to plug the open blood vessels so that you don't have excessive bleeding. We have ion recycling. During the breakdown of red blood cells, we have the hemoglobin, which contains iron. The iron is recycled back into the bone marrow. So this marrow, not wrong spelling here, the bone marrow, where that can be reused during the process of hematopoiesis. We also have the spleen acting as an immunological role by production of antibodies. The B cells are exposed to the antigen by antibody-presenting cells. These B cells then, after being exposed or activated, are transformed to plasma cells, which then can get into the tissues where you can have infection occurring. We also have within the splenic cords, within the white pulp, we have facilitation of conduct of B and T lymphocytes with the macrophages. These macrophages are antigen-presenting cells. We also have other antigen-presenting cells that are referred as dendritic cells. Send the antigen to the B cells so that they can be activated. The B cells can further be activated by the T helper cells within the splenic cords. We also have sequestration of monocytes and transformation into macrophages. Remember that monocytes are in circulation. They must get out of circulation. They then transform to become macrophages, which are larger than the normal monocytes. These macrophages are removed from the circulation as monocytes into the spleen as macrophages, and then they can invade other tissues, and that is important. Hematopoiesis in the fetus, we have during fetal stage, we have the spleen acting as part of the hematopoietic organ, just like the liver during the mesoblastic stage and also the hepatic stage. That is where we have hematopoiesis taking place, and spleen plays a role in this. This is a global view of the spleen. In this global view of the spleen, when you take a fresh spleen and cut it, I've said you see some white spots and also some red areas. These white spots are areas which we refer to as the white pulp. They consist of aggregates of white blood cells, the lymphocytes. When we stain the spleen, this is what we see as the red pulp, white pulp, comprising of aggregates of lymphocytes, both T lymphocytes and B lymphocytes, and then the rest of the areas are the red pulp, comprising of splenic cords of Billroth and also the splenic sinuses. They appear red because we have a lot of blood in these splenic sinuses, and that's why we know that the spleen acts as a reservoir of blood. So what is the global structure of the spleen? We normally have a capsule, which is fibrous connective tissue, but the spleen also contains smooth muscle. That smooth muscle is very important in contracting to squeeze out the blood into circulation if we have excessive bleeding. That's why we consider the spleen as a reservoir. This capsule penetrates into the organ as trabecula. This trabecula consists of fibrous connective tissue and smooth muscle. That divides the spleen into lobules, and the individual cells of the spleen are then supported by reticular fibers. As the blood vessels enter the spleen, we have the trabecular artery, and this trabecular artery then branch off to give a central arteriole. This central arteriole is surrounded by the white pulp. And the central arteriole, you will note from this, is not at the center, is eccentric. It's not at the center of this called central arteriole, but is eccentric. And this is surrounded by the white pulp. The white pulp surrounding the central arteriole is referred to as the periarterial lymphoid sheath. It's acting like a sheath because it is surrounding the blood vessel. If you know the structure of the blood vessel from what you have done, the blood vessel contains of tunica intima, tunica media, and tunica adventitia. The adventitia of the central arteriole comprises of the periarterial lymphoid sheath, which is mainly T lymphocytes. So the lymphocytes surround the central arteriole. We are going to look into the details. The white pulp also comprises of splenic nodules. And if you look at this nodule, it's typical of what you will see of a follicular nodule, comprising, if it has been activated to antigens, comprising to a central germinocenter and a cape or a mantle zone peripheral. A central germinocenter and a cape or mantle zone peripheral. So this is typical of what we had described earlier. So we said the spleen, grossly, I think the gross people will tell you that it's attached to some organs, attached to the stomach by gastrosplenic ligament, attached to the liver and the kidney by a lienorenal ligament, attached to the phrenic, the diaphragm, attached to the intestines. So these are ligaments that attach the spleen and therefore keep it in position. Some of the malformations of the spleen originate from the mesoderm, and some of the malformations that can arise will be some accessory spleen, a splenic tissue, or sometimes no spleen develops, or a very small spleen can develop, or a very large spleen can develop. That's hypersplenia. And sometimes during injuries, the spleen can be removed by the process of splenectomy. Because it's a tissue that tends to rupture if there is an injury, there can be excessive bleeding, and therefore it has to be removed. And then, like I said, the stroma consists of the capsule, dense fibrous connective tissue, the trabecula extending from the capsule, comprising of smooth muscle and collagen fibers, and then finally the reticular fibers, providing the individual supportive framework for the cells. And then the parenchyma, that's the cells of the spleen. We look at the parenchyma as comprising of the white pulp, which appear grayish-white, but in a freshly cut spleen, it appears as white spots because it comprises of aggregates of white blood cells. Then we have the periarterial lymphoid sheath, which I've talked about, surrounding the central arteriole. This periarterial lymphoid sheath comprises of T lymphocytes, and this, I've said, replaces the tunica adventitia of the blood vessels. And then around that periarterial lymphoid sheath, we also have macrophages. We have macrophages, and these macrophages are important in presenting the antigen to the T cells so that they get activated. Sometimes we have absent spleen in what we call DiGeorge syndrome, and this is mainly phase one, the pharyngeal arches development, but also involves the spleen as part of the syndrome of the DiGeorge syndrome. Because the thymus as a problem, we therefore have the periarterial lymphoid sheath lacking in the spleen. That's why we are relating the absence of the thymus to the spleen in terms of the periarterial lymphoid sheath comprised of T lymphocytes. If there is an absent thymus, then that area of the periarterial lymphoid sheath will be lacking. The parenchyma also comprises of splenic follicles, and like we had said, the splenic follicles or nodules consist of aggregates of cells, and these aggregates of cells are scattered among the periarterial lymphoid sheath. The splenic follicles will be having lymphocytes, both P lymphocytes stage. It will be having macrophages, which are very important, and also dendritic follicular dendritic cells are very important, as they're very important, playing a role as antigen presenting cells. This presents the cells to the B lymphocytes and also to the T lymphocytes. In the primary, there is no germinal center. There's no germinal center. So we do not have the activation in the case of the primary. In the secondary splenic nodule, which are just shown, there is a germinal center. And this is where antigens have been presented to the T lymphocytes and the B lymphocytes, and there is activation of the cells. So those immunoblasts are going to multiply, and they are also going to form memory cells. So the cap, the outer part of the follicle, will containing some B lymphocytes which have not been exposed to the antigen, whereas the central part comprises of B lymphocytes that have been exposed to the antigen. We also have the plasma cells. These are cells which have been exposed to the antigen that will now get into circulation. And of course, the macrophages are important. Now, the germinal center comprised of the B lymphocytes that are not undergoing division, that are in resting stage, and also B immunoblasts. These are cells which are actively dividing. These B immunoblasts will have been activated from the B lymphocytes which are resting by the dendritic cells that are acting as antigen-presenting cells. But also the T helper cells are very important in the maturation of the B lymphocytes to form the plasma cells. Plasma cells are those that are producing antibodies. So we need to know that it is not the naive B lymphocytes that are producing antibodies, but it is the plasma cells that are producing antibodies. In the splenic follicle, that's the nodule, the marginal area. Remember, we have the germinal center, we have the mantle, and then we have the marginal zone. The marginal zone is the junction to the red part. What activity do we have in the marginal zone? The marginal zone contains reticular cells. If you remember, reticular cells are cells that produce reticular fibers. But we also have dendritic cells, which are very important in activation of the cells as antigen-presenting cells. We have B memory cells within the marginal zone, and we have macrophages. The macrophages are around the splenic follicle, but macrophages are also at the edge of the periarterial lymphoid sheath. And like I've said, macrophages are very important as antigen-presenting cells. So the marginal zone is actually the area where we have the presentation of antigens to the B lymphocytes and the T lymphocytes so that they can be activated. So it is the area where we have induction of immune response within the marginal zone. At the marginal zone, we also have marginal sinuses, and marginal sinuses contain blood, and this is the zone where the cells now migrate into circulation. When they migrate into circulation, these cells contain specialized type of endothelial cells that we refer to as staph cells. Staph cells are long cells, and their longitudinal axis is, they of course are endothelial cells, which are, unlike the flattened endothelial cells, these ones are not flattened endothelial cells. The longitudinal axis of the nuclei is parallel to the endothelial basement membrane, to the endothelial basement membrane. So this is unique. The staph cells are also unique. These staph cells have gaps in between the cells that are very important in facilitating the migration of cells into circulation. Migration of cells into circulation. Therefore, those cells which are deformed, like the red blood cells which are deformed, will not be allowed into circulation. Destroyed, will be destroyed before the cells get into circulation. The normal cells will get, red blood cells will get back into circulation. The rest will be destroyed. This is a splenic nodule. This is the germinal center. This is the central arteriole. We have said is eccentric. This is the cape or the mantle. And this is the marginal zone. The marginal zone. So it is in this marginal zone that we have the activation. It's also in this marginal zone that we have the cells migrating back now into circulation. The mature cells, the plasma cells, can migrate into circulation in this particular area. Then we have the red pulp. Remember the parenchyma we have said comprised of the white pulp, which consists of B and T lymphocytes primarily, and also the antigen-presenting cells that comprise of dendritic cells, follicular dendritic cells, and also macrophages. Within the red pulp is the majority of the in volume, still comprising 2/3, and the red pulp comprises of splenic cords of bilirubin, and also splenic sinuses. The splenic cords comprise of reticular cells. Remember, we said most of the supportive framework are reticular cells that produce reticular fibers. We also have dendritic cells, which function we have talked about as antigen-presenting cells, macrophages as antigen-presenting cells. We have the monocytes which have been sequestered We said spleen acts as an area for sequestration of monocytes and conversion of monocytes to macrophages. Then we have the B lymphocytes within these splenic cords. So the splenic cords is an extension of the white pulp. And the splenic cords primarily function as areas of filtration of blood, or what we talked about earlier as blood quality regulation. And this filtration, in the filtration area, is also removal of bacteria, removal of viruses, removal of foreign proteins, removal of blood-borne antigens by the macrophages and other antigen-presenting cells, phagocytic cells. Then we have the splenic sinusoids. The splenic sinusoids comprise of large channels of blood vessels that are lined by staph cells, which we talked about. These staph cells are rod-shaped cells, unlike the typical squamous cells of the endothelium. These rod-shaped cells have gaps in between them that are allowing migration of cells. So it is selective passage of cells. The endothelial cells are surrounded by circularly arranged reticular fibers and adventitial cells. These circularly arranged reticular fibers and adventitial cells act as filtration barriers. So they also help in selection of cells that can be able to pass through into the sinuses. The endothelial clefts are present, no basement membrane of the sinusoidal capillaries. We have no basement membrane. Remember that sinusoidal capillaries have no basement membrane, and that is very important. And this is to allow for passage of red blood cells and also other wandering cells into circulation. And out of circulation into the filtration fields. We are going to talk about blood flow, and that is important. This is the blood sinuses. These are the staph cells. They are rod-shaped. This is the lumen of the blood vessel, the sinus, with the normal blood cells. Like these ones are the normal neutrophils, basophils, eosinophils. The normal formed elements of blood will be in these areas. These cells can get out into these areas that are part of the so with this area you can have the filtration of blood. We had looked at this picture before. Blood enters as splenic artery, breaks down as penetrates into the organ as trabecular artery. From trabecular artery, this branch off to form the central artery that we had said is eccentric. The central arteries breaks out into splenic, the peninsular arteries. And then in the area, the junction between the red pulp and the white pulp, the peninsular arteries break down into sheath capillaries. We have two methods of blood flow. We have the first method of blood flow. In this continuous method, blood flows entirely, just like what normally in the normal circulation. So the continuous we also in from the start 2 to 6 and then the capillaries as blood flow into the sinuses, splenic sinuses. So by contraction of these smooth muscle areas around the shift capillaries, we have less blood flowing into the open circulation, and therefore more blood is channeled through the closed circulation. So up to this part here, this is what we refer to as the closed system of circulation. From the splenic sinuses, capillaries, blood then flows into splenic sinuses. It bypasses the splenic cords of Bellroth. That we refer to as the closing of circulation, in which blood is entirely surrounded by endothelia. And then in the open system of circulation, blood flows from shift capillaries into the splenic cords of Bellroth. We have talked about what the splenic cords of Bellroth contain. We have talked about the functions of the splenic cords of Bellroth as areas of filtration. And then from the splenic cords of Bellroth, blood gets back into the splenic sinuses. Because remember, the splenic sinuses are lined by stem cells, which allow blood to flow back into circulation. So the red blood cells, which are deformed, will have been destroyed within the splenic cords of Bellroth. The normal blood cell, red blood cells, which have not been deformed, can squeeze back into the splenic sinuses and continue into circulation. Then we have the pulp vein, trabecular vein, then splenic vein, and then usually the inferior vena cava. So this just to illustrate the blood flow as trabecular artery giving rise to central arteriole, and the central arteriole is surrounded by periarterial lymphoid sheath, which is a thymus dependent zone because it contains T lymphocytes. And then from there we have the central artery. The central artery again surrounded by periarterial lymphoid sheath, and from there we have the peninsular arteries, which gives rise to the sheathed capillaries. The sheathed capillaries. The sheathed capillaries then enter back into sinusoidal capillaries. These are the splenic sinuses. So note that in this position, it's continuous, entering into splenic sinuses. In this position, it's opening into the splenic cords of Bellroth. Splenic cords of Bellroth, where it is filtered and then enters back into the splenic sinuses. So this is the open circulation. This is the closed circulation. Remember that in the closed circulation, blood flows entirely surrounded by endothelia. And then after the splenic sinuses, it gets back into the venules, So just to recap, because it's important, for open circulation, blood percolates through the splenic cords of Bellroth, where maximum filtration takes place. For closed circulation, blood bypasses the splenic cords of Bell and continue directly into the splenic sinuses. So the blood flow bypass splenic cords directly into the splenic sinuses. So splenic cords of Bell maximize on filtration. That is what we have as the structure of the spleen, which is the largest lymphoid organ. Again, let's remember two things. In the case of the bone marrow, bone marrow receives blood. Thymus receives blood. So all that blood is received. In the case of the thymus, acquisition of receptors, so it teaches the T lymphocytes to recognize foreign antigens and foreign proteins. Then from there, the blood flows to the thymus-dependent zones. Insulin, periarterial lymph is a thymus-dependent zone. Uh, uh, lymph is a get out of circulation. So once the fluids get out of circulation, the cells are bathed by fluid. The interstitial fluid is very rich in oxygen, very rich in nutrients, rich in oxygen, rich in nutrients. Blood, the fluid, then must get back into circulation. In the interstitial space, we also have wandering cells. Wandering cells, because we have tissue injury, wandering cells are very important in immune surveillance. So these cells must get back into circulation. To get back into circulation, they enter into the lymphatic capillaries. Lymphocytes enter into lymphatic capillaries. Again, the fluid within the lymphatic capillaries, the lymphatic fluid must be cleaned. So lymph tissue is picking up lymphatic fluid containing cells, might be containing foreign bacteria, containing viruses from the tissues after injury, containing foreign proteins, capillaries, then by passing through a lymph nodule bed. Lymph node, and it's very important for antigen presenting lymph nodes also are in recirculation of lymphocytes. The lymphocytes that are passed into the tissues, lymphocytes get back into the lymph node. Lymph nodes produce activated lymphocytes. pass the lymphatic study of lymph nodes. Lymph nodes. Remember that we have popliteal region just behind your knee joint, and then after that, the and then swelling, are so they are able to clean the lymph before the lymph gets back into circulation. Before the lymph gets back to circulation. Duncan and Korir, please mute your microphones and let me not remind you again. Alex, mute your microphone. So I'd like you to pay attention to these regions because usually when there's an injury, these are the lymph nodes which swell, and therefore you are able to note that there has been an injury. What would be the function of lymph nodes? We have said the lymph nodes are organized in a series. So lymph flows from one lymph node, and from that lymph node, it passes to another series of lymph nodes before it gets into circulation. So these are able to filter and remove all foreign material before the lymph gets back into blood. So the plasma is returned back into blood as lymphatic fluid. So the function of lymph nodes is filtration. Filtration by phagocytosis. This phagocytosis can be by macrophages, by free macrophages, macrophages within the lymph nodes, and also reticular cells are capable of phagocytosis. Immune surveillance. So they monitor the lymphatic fluid as it passes through, and in the process picks up any antigens and presents it to the B lymphocytes and the T lymphocytes so that those can be activated, either to remove the bacteria, to kill the bacteria, to kill the viruses, or to produce antibodies against the foreign material. So most of these contain macrophages and dendritic cells, which capture the antigen and present it to the T lymphocytes. Within the lymph nodes, we also have the T helper cells, which help in the maturation of the T lymphocytes, just like what we had mentioned in the case of the spleen. It's also sites for immune cell activation, activation of the P lymphocytes from plasma cells, which produce antibodies, and also activation of the T cells. We have areas that we refer to as thymus dependent zone, which contain T cells. And very important, recirculation of lymphocytes. So what is the structure of a lymph node? Just like any other solid organ, it's covered by a connective tissue capsule. The capsule penetrates into the organ as trabecular, and the organ itself is supported, the cells themselves are supported by reticular fibers. We note that the lymph node contains a cortex, the outer part, and the cortex is divided into two parts. An outer cortex, which contains lymph nodules, typical of the lymph nodule, either primary lymph nodule or secondary lymph. Primary lymph nodule before they're exposed to antigens, secondary lymph nodules when they're exposed to antigens, and the secondary lymph nodule contain the germinal center. So within the lymph nodule, we have the mantle zone, remember? We have the mantle zone, the germinal center, where we have the T helper cells that are very important in maturation of the B lymphocytes. We also have follicular dendritic cells. And the follicular dendritic cells, like we had said, are very important as antigen presenting cells. Within the germinal center, we have light zones and dark zones, light regions and dark regions. Also, in the germinal center, we have no major histocompatibility expression, and this is in the primary outer part. Then, once the antigens have been collected and presented to the primary, to the B lymphocytes, with the help of T lymphocytes, there is then activation. Then we have the secondary lymph nodule. Then we have the inner parts, inner cortex. The inner cortex comprised of diffuse distribution of lymphocytes. Diffuse distribution of lymphocytes. It also has follicular dendritic cells, and unlike what we have in the cortex, these follicular dendritic cells have more processes. They have more processes, fewer organelles, and irregular nucleus. This way of major histocompatibility expression. In the inner cortex, the endothelium is thick and contain what you refer to as high endothelial veins. High endothelial veins. The high endothelial veins are very important in allowing migration of the lymphocytes. Migration of lymphocytes back into circulation. That's why we say that the lymph node is very important in recirculation of lymphocytes. So this is a global view of the lymph node. We have a capsule, and within the capsule, this is fibrous connective tissue, penetrate into the organ as trabecula. Entering at the capsule, we have the afferent arterioles, the afferent lymphatic vessels. So afferent lymphatic vessels enter capsule at the cortex. Within the cortex, the capsule, we have the capsular sinuses, and the capsular sinuses divide to form trabecular sinuses. So the trabecular sinuses accompany the trabecula. And that's why we say that the mantle zone are the areas of the sinuses, trabecular sinuses are thickest. This is the outer cortex comprising of nodules, and once they have been activated, we have a germinal center and the mantle zone, which we had talked about earlier on. This part here is the paracortex, and paracortex is a thymus-dependent zone. It contains T lymphocytes. It also contains high endothelial veins. Postcapillary veins have high endothelial veins that help in the migration of the cells back into circulation. So what enters at the cortex is lymph. Once that lymph gets into the cortex, it passes into the medulla as medullary sinuses. So we have capsular sinuses, subcapsular sinuses, trabecular sinuses, and from here it then enters through these areas where most of the filtration takes place, and the lymph eventually enters as medullary sinuses. We have the medulla. Within the medulla, we have medullary sinuses and medullary cords. These are medullary cords, and medullary cords are extensions of the paracortex, and they contain plasma cells. They also contain macrophages, molecular dendritic cells within the medullary cords. So these are medullary cords and medullary sinuses forming the medulla. This is a nodule at the outer part, and the nodule, remember, once exposed to the antigen, have the germinal center and then the mantle zone or the cape. And the mantle zone is thick, close to the trabecular sinus. Notice also that within the germinal center, we have the light areas and the dark areas. Dark areas containing closely packed immunoblasts. So this is what we have as the lymph node. And for the lymph, it enters as afferent lymphatic vessel, which breaks down to capsular sinuses. The capsular sinuses form cortical sinuses, which form subcortical sinus, and subcortical sinus form peritrabecular sinuses. From the peritrabecular sinuses, we have a very large filtration zone. And this filtration zone contains reticular cells, macrophages, Langerhans cells, mast cells. So remember, this is the area where we have antigen presentation. This is the area where we have the removal of bacteria, removal of cells by both reticular cells and also mast cells and stellate cells. Now, the endothelia here, remember, is unique also in a sense that it contains the high endothelial cells. These stellate cells have a lot of processes. That's why we refer to them as stellate endothelial cells that are almost columnar instead of being squamous endothelial lining. Then the lymph gets into the medullary sinuses. From the medullary sinuses, all the lymph collects as efferent lymphatic vessels. So you can have either one or two efferent lymphatic vessels, but several afferents enter in at the convex part of the lymph node. So the efferent leave a hilum of lymph node. At the hilum of the lymph node, we also have blood vessels entering. We also have nerves entering at the hilum of the blood vessels, at the hilum of the lymphatic lymph node. So this is to show lymph flow, afferent lymphatic vessels, and the efferent lymphatic vessels leaving. The cortex, the lymph nodules and follicle, paracortex with the diffuse lymphoid tissue, and then the medulla with the medullary cords and medullary sinuses. From the medullary sinuses, all the lymph, once it has been filtered, then leaves as efferent lymphatic vessel. All these lymphatic vessels are equipped with valves to ensure unidirectional flow of lymph. We say the lymph nodes are arranged in a series. So this efferent lymphatic vessel becomes an afferent lymphatic vessel for the next lymph node. And the process continues on until the lymph enters into circulation. Within the abdominal cavity, all the lymph collects to form the cisterna chyli, and this cisterna chyli eventually empty into the thoracic duct, and the thoracic duct then open into the jugular vein, so all the lymph enter back into recirculation. At the hilum, this is the area that we refer to as the hilum. This is where we also have the blood vessels enter to supply oxygen and nutrients to the cells of the lymph node. At the hilum, we also have the veins living at the hilum. These are removing the carbon dioxide and also waste products from the tissues of the lymph node. So remember that unlike the spleen, unlike the lymph node, what flows here for the purpose of immune surveillance and cleaning is lymph. The blood vessels are just to supply the tissues with the nutrients and oxygen and to remove the metabolites. This is a capsule with a capsular sinus. We have subcapsular sinuses. Remember that all the lymphatic vessels have valves. And then from the subcapsular, we have the trabecular sinuses. So that is about a lymph node. The other secondary lymph nodules, lymphatic, are the tonsils. Remember, when we talk about lymphatic tissue, we are saying that they contain aggregates of B lymphocytes and T lymphocytes. So tonsils contain B lymphocytes and T lymphocytes. The distribution of this lymphoid tissue is the same. We have either as a lymph nodule, which are aggregates that are either oval or diffuse lymphoid distribution, in which case the lymphocytes are uniformly distributed without any sequestration. So let's now look at the tonsils. Tonsils are usually located in our pharyngeal area, and pharyngeal area is a very important area. It's an area where air passes through into the lungs, where food passes through into the esophagus. It is also an area where there is an opening of the nasal cavity. It's also an area where there is an opening into the middle ear through the pharyngotympanic tube, also referred to as eustachian tube. Need to be able to have more lymphoid tissue as a protective mechanism. Within the pharyngeal area, the tonsils include what you call palatine tonsil in the palatal fossa, with palatoglossal fold and palatopharyngeal fold. That's the palatal fossa. The lingual tonsil at the base of the tongue, and the pharyngeal tonsils at the roof of the pharynx, which also will include lymphoid tissue within the uvula. The pharyngeal will also include lymphoid tissue that guard the opening of eustachian tube or the pharyngotympanic tube, what we refer to as the tubal tonsil. What will be the characteristic of the tonsils? One, it's associated with epithelia. So in the case of the palatine and the lingual tonsil, we have stratified squamous epithelia. Stratified squamous epithelia. In the case of the pharyngeal tonsils, at the nasopharynx, we have extension from the nasal cavity. So we have pseudostratified ciliated columnar epithelia as the lining. Immediately below the epithelia, in the lamina propria, we have the lymphoid tissue. And within the epithelial areas, we have a lot of crypts, a lot of furrows, crypts. These crypts are very well developed in the palatine tonsil, and they also appear branched. That is important, well developed and branched. Within these crypts, we have squamated cells. We also have some debris. And therefore, these crypts allow for adequate time for exposure for the antigen presenting cells to pick up the antigens and present them to the B lymphocytes and the T lymphocytes. The lingual tonsil do not have very well, and the pharyngeal tonsils have it. We said that organs contain capsule, but for the tonsils, these are lined on only one side. The epithelium is lined on one side, so the capsule is incomplete. So there's a partial capsule that separates it from the underlying tissues. In the case of the palatine tonsil, we do not have any distinct capsule in the case of the lingual tonsil, and we might have complete capsule that is patchy in the case of lingual tonsils. The location, palatine fossa, the base of the tongue, the what we have the lymphoid tissue, and this lymphoid tissue will consist of either follicles, which is very nice oval spherical aggregates of lymphocytes, or diffuse. Between the follicles at the interfollicular area, in the case of the palatine tonsils, we have T cells with high endothelial veins. High endothelial veins will allow for migration of the cells. And also the dendritic cells are present for presentation of the antigens to the B cells and the T lymphocytes. So these are comparisons of the tonsils. Within the pharynx area, the tonsils form what we call the Welder's ring. And the Weldeas ring comprises of the palatine tonsil, the lingual tonsil, the tubal tonsil, which is part of the pharyngeal tonsil, but surrounds the Australian tube, so that forms the Weldeas ring, heading into the respiratory tract, into the digestive tract, and also into the milia. We also have a part of the mucosal tissue that is distributed in many other areas. So we refer to this as mucosal associated lymphoid tissue. Remember, when you talk about mucosal associated lymphoid tissue, this is associated with the mucous membranes. A mucous membrane is a lining that opens to the outside. Mucosal associated lymphoid tissue include gastrointestinal associated lymphoid tissue that is associated with the digestive tract. Of importance in the gastrointestinal lymphoid tissue is Peyer's patches. Peyer's patches in the ileum, and within the appendix, the entire lamina propria and the submucosa. So the ileum contains Peyer's patches, and these Peyer's patches are aggregate located in the submucosa of the ileum at the antimesenteric part of the submucosa, and also sometimes in the lamina propria. Then we have the bronchial associated lymphoid tissue associated with the respiratory tract within the bronchi. We have a lot of isolated lymphoid tissue. And the adenoids. This is nasal associated lymphoid tissue, and typically what we call the mucosal associated lymphoid tissue. And these are distributed times of the salivary glands, around the breast, around the lacrimal and the thyroid areas might have some associated lymphoid tissue. Tonsils are part of the mucosal associated lymphoid, and I talked about components of the Weldeas ring. Components of the Weldeas ring. So remember the uvula, that part that hangs from the most posterior part, if you open your mouth, in the soft palate that is referred to as the uvula, also contains some lymphoid tissue that sometimes gets enlarged, especially in children. Then this is cut off. Associated with tissue tonsils. First, we have stratified squamous non-keratinized epithelium, or it might be like in the pharynx, stratified. Then we have lymphoid tissue below the basement membrane, and sometimes the lymphoid tissue are also invading into the epithelia. And when they invade into the epithelium, we refer to it sometimes as intraepithelial. lymphoid in lymphoid. Yeah. Complete capsule. In complete capsule. So that a lot of branches. So palatine is the one normally which is prone to this or and also issue exposure and postulation of the lymph tissue. That's why we refer to it as bronchial associated lymph, where we have a lot of this lymph tissue cross the epithelium. The epithelial cells will have, especially in the senile part, will have M cells. The M cells, the E cells, which capture the antigens, T cells and the B cells. B cells are part of the line of the intestines where we have lymphoid tissue. And their purpose is to capture the antigen and present it to the B lymphocytes and also T lymphocytes. So this is our part is that in the lamina propria, we have lymphatic lymphocytes, and also of the appendix in the lamina propria. This is the ileum, and in this ileum, notice lymphoid tissue in the lamina propria, lymphoid tissue in the submucous, lymph nodes, and also diffuse tissue. And this is only on the one half. That's why we say it's only in the antimesenteric part of it. So that is about our lymphoid, and that concludes our session. Remember, we talked about the thymus, recognize the septum recognize the cortex of the thymus, recognize the medulla, forming an incomplete medulla, unlike what we have in the case of the lymph node. Recognize also the type of cells that we have. In the case of the bone marrow, hematopoietic compartments, and the vascular compartment. Recognize Hazel's corpuscles here, comprising of concentric that can undergo keratinization. So this is very typical of the medulla of a thymus. Also note that thymus can undergo involution, where thymic tissue is completely reduced and we have replaced by adipose tissue. This white, so these are just remnant tissue. Hazel's corpuscle. Note the thymocytes and note these are the red cells here, reticular cells, thymocytes and reticular cells. This is in the formation of a Hazel's corpuscle. Is this clear? Capsular, subcapsular sinus, follicle, lymph follicle, primary lymph follicle, diffuse lymphoid, capsule containing collagen fibers, adipose tissue that surround the organs, lymph nodes, outer cortex, inner cortex containing T lymphocytes and high endothelial veins, medulla containing medullary sinuses and medullary cords. So that these are divided. This is just a capsule showing the endothelial lining of the sinuses. And this one here are reticular cells that produce reticular fibers. This is the reticular fibers. Notice the network formed by reticular fibers for the reticuloendothelial organs. This, I think, is familiar, the palatine tonsils. Hazel's corpuscle at high power. The spleen, notice the splenic nodule, the trabeculum, compressive smooth muscle and collagen fibers. Eccentrical central vein, smooth muscle and also collagen fibers for the spleen. So note the capsule having smooth muscle unlike other organs. The trabeculae also having smooth muscle. Note the eccentrically placed central vein and very clear nodule. Identify the capsule, identify the red part, identify the trabecula, identify the central vein, identify reticular cells, and the others are lymphocytes. Central vein, periarterial lymphoid sheath, and then these other areas also containing the splenic cords of Billroth and the splenic sinuses. For the blood cells, I think not the monocyte, not the basophil, not the biconcave red blood cells, and this shape is maintained by spectrin, which confers flexibility that is what I now invite any questions. If you have any questions, please put them in the chat.